Tonight on Nova. 10-month-old Laura has cancer, but this houseplant contains a chemical that may save her life. From everyday painkillers to prescription heart medications, modern pharmacies are stocked with powerful drugs that were first derived from plants. Scientists are searching for new medicines among the exotic plants of the tropical rainforest. But will they find tomorrow's wonder drugs before the forest disappears? Tonight on Nova, the hidden power of plants.
Major funding for Nova is provided by this station and other public television stations nationwide. Additional funding was provided by the Johnson & Johnson family of companies, supplying healthcare products worldwide. And by Allied Signal, a technology leader in aerospace, electronics, automotive products, and engineered materials.
By any measure, the plants of the tropical rainforest are beautiful and mysterious. In this profusion of leaves and blooms are thousands of plant species, many still undiscovered and unnamed. Plants that provide shelter and nourishment for the animals that live among them. Plants that help sustain the Earth's atmosphere, renewing the oxygen that makes life possible. And plants that provide medicine. Indeed, the history of medicine has been largely the story of plants and the potent chemicals they produce. Around the world, traditional healers using plant medications provide health care to 80% of the human population, over 4 billion people. Ayurveda, also called the science of life, developed in India more than 3,000 years ago, laying the foundations for one of history's most remarkable medical traditions. This system of herbal remedies and natural healing is still dominant in India. There are more than 8,000 prescriptions in this ancient system of medicine, and factories like this one process raw materials into preparations used by over 500 million people. From Ayurveda, the Western world learned about Rauwolfia serpentina, or the snakeroot plant. Also known as reserpine, it is used to control hypertension and is a compound essential to modern cardiology. Like India, China is the home of an ancient and sophisticated natural medicine system. Over 4,000 years, the Chinese have discovered, developed, and refined more than 11,000 medical prescriptions based on the natural chemistry of plants. These mixtures and compounds were based on centuries of empirical observation of their effects on the human body. Using ancient recipes, remedies are carefully weighed and measured in accordance with the physician's requests. While the final prescription may contain unusual-looking elements, the herbal drugstore is as common in China as the corner drugstore in the West. Even in parts of the world where vegetation is sparse, there is a legacy of medicinal plants. For centuries, the Bedouin have used ami seeds, which contain cromolyn, as an effective asthma remedy. Even our own Western pharmacology, with its pills and white crystalline powders, owes much of its potency to the plant kingdom. Many common medications were first derived from plants. More than 2,400 years ago, the Greeks used extracts of willow bark to treat pain. The active compound it contained is better known today as aspirin. The flowering foxglove was used to treat heart problems as far back as the 15th century. Today, digitalis from the foxglove is still an important component of heart medications. Aspirin, digitalis, and many other medicines are now synthetics, chemical versions of plant compounds. But not all modern medicines can be synthesized. Over 25% of the drugs prescribed in the U.S. still contain plant materials as their principal active ingredient.
According to a recent study by a congressional group, the U.S. imported over $24 million a year of medicinal plants in 1974 and then turned these into $3 billion worth of medicines. So it is fairly a significant business.
What is it about plants that gives them the power to heal or to harm? For in addition to their beneficial uses, some plants are well known to contain chemicals that are harmful to the human body. Rhubarb leaves, for instance, produce a deadly broth of chemicals that can cause diarrhea, vomiting, and can prevent the blood from clotting. Every part of the potato plant, except the fleshy tuber, can cause severe poisoning. What lies behind the mysterious chemistry of plants? Of all living things, only plants have the power to transform light energy into life energy. This precise chemical process is known as photosynthesis. These leaf cells under the microscope reveal a constant stream of green particles called chloroplasts. Magnified 40,000 times, the inside of the chloroplast is seen to be made up of tiny light collectors arranged in layers. There are thousands of such chloroplasts in each green leaf, performing the task of photosynthesis. Within the leaf, carbon dioxide absorbed from the atmosphere and water taken up by the root system undergo complex chemical reactions fueled by energy from sunlight. The result is the simple sugar glucose, which is stored in the leaf cells as food. The oxygen that is released back into the atmosphere is simply a byproduct of the process. The primary products of photosynthesis, carbohydrates and sugars, sustain the plant itself, and the plant can then in turn be consumed as part of the food chain. Yet only a few of the many chemicals produced by plants are primary compounds. Another group of chemicals called secondary compounds are unusual substances that do not seem essential to the plant's basic life cycle. For the most part, the functions served by these secondary compounds are still unknown. Why does oleander manufacture cardioactive glycosides that can paralyze the lungs, cause bleeding, and even death? What is the role of calcium oxalate in household caladiums? In humans, it irritates the throat and can lead to asphyxiation. And why does belladonna produce hallucinogenic compounds? One view is that the plants need these substances for protection and defense.
It comes back to the competition between living organisms for nutrients. And for example, if you look at the tropical areas, you have a tremendous variety of plants, and all of them have everything they need to grow very, very prolifically. They have oxygen, they have light, they have soil, warm temperature, everything except space. And the limitations of living space force these plants to prepare and to secrete different sorts of protective substances which enhance their ability to survive, either by preventing predation or preventing attack by insects, or by enhancing the destruction or limiting the ability of other plants to grow around them.
But scientists are not in complete agreement about the role of secondary compounds. For some, they remain mysterious entities.
We don't know what their function is in plants. We have absolutely no idea. And a lot of people are inclined to dismiss this as being idle metabolic chatter on the part of the plant, for example. Some useless chemical. The plant doesn't know what it's doing, so it spends a lot of its time dreaming up some scheme for making weird molecules, which people like myself might be interested in, you know.
Whatever purpose they may serve in the plant, these secondary compounds are the critical components of plant medicines for human use. And the search for hidden power in plants has often revolutionized medicine. In 1803, a young German pharmacist, Friedrich Turner, was looking for the active substance contained in opium from the poppy, whose good and evil influence had been known for centuries. He was the first to identify an alkaloid, a secondary plant product characterized by its molecular structure, which includes at least one nitrogen atom, and by its ability to affect the human body. Once the alkaloid had been isolated, Sir Turner could refine the opium into a more powerful drug, with more precise action in the body. After testing the new drug on himself and his colleagues, he named it morphine, for Morpheus, the Greek god of sleep, and its pain-killing properties remain unsurpassed. Alkaloids are the active compounds in such familiar drugs as nicotine, caffeine, and cocaine. They have also accounted for modern-day medical breakthroughs. The Madagascar periwinkle has a long history as a traditional remedy, used to treat everything from sore throats to diabetes. In the 1950s, scientists discovered that an alkaloid called vincristine, produced by the periwinkle, had a new application in treating childhood leukemia and other cancers. 10-month-old Laura is receiving vincristine as part of a program of therapy to stop the growth of a malignant tumor. Her physician, Dr. Mark Greenberg, describes how vincristine works.
Vincristine is a mitotic spindle poison. What the drug actually does is to arrest the division of cells. When a cell of any sort divides, its chromosomes split into two and then attach themselves to what's called a mitotic spindle. And the mitotic spindle helps to pull the two halves of the chromosomes apart. What vincristine actually does biologically is to poison that spindle, or the tubules that form that spindle. It does this preferentially with malignant cells. And so what it's doing is really attaching to any tubules, any mitotic tubules in malignant cells that are in the process of dividing.
The use of vincristine is just one part of an aggressive medical approach to childhood cancer.
We almost never use drugs by themselves. We use a combination of drugs that act at various points in the cell cycle. So that vincristine, used in combination with many other drugs, is enormously effective in this sort of range of malignant disease in childhood.
Between the new drugs and the new methods of chemotherapy, we've gone from what was perhaps 10 or 15% 5-year survival in childhood leukemias, now up above 85 or 90%.
Other life-saving medical breakthroughs have recently come from the natural world. A vacation stroll in Norway 15 years ago led to a discovery that revolutionized the science of organ transplantation. employee of a Swiss drug company looking for dirt collected a few spoonfuls of mud from a murky swamp. He brought the mud back to the lab here in Basel, Switzerland. Because many antibiotics have been discovered in soil, drug companies routinely check soil samples from all over the world to see what they contain. Among the several microbes which grew from the Norwegian mud was a fungus that had never been seen before. This new fungus produced a chemical that had an unexpected benefit. It could counteract organ rejection. Although the surgical techniques that allow transplants have been in use since the first kidney transplant in 1954, the transplants themselves were only moderately successful. In order for transplants to work, a significant problem must be overcome. A transplanted organ is perceived as a foreign invader by the body's white blood cells, which then attack the new organ, in this case a kidney. This immune response overwhelms the organ and leads to rejection, unless the recipient is given immunosuppressant drugs. These drugs kill the white cells that cause rejection, but they also kill all the other cells that protect the body from intruders, leaving the patient vulnerable to serious infection. More than ever before, cyclosporin solved the problem. When the white blood cells start to attack a new organ, cyclosporin calls them off. But unlike the other anti-rejection drugs, it doesn't kill them. It just stops their assault. So the organ is protected, but the white blood cells can still patrol the body and keep it free from infection.
So we go right at the joint line right above.
Over 7,000 transplants are performed in the U.S. each year. Since cyclosporin was first made available in 1983, the success rate of certain transplants has grown to nearly 80%. The recent discovery of such drugs as cyclosporin and vincristine suggests that there is still untapped potential in nature's chemistry and has inspired the search for new medical products from plants.
When you consider that all the drugs that come from plants in the world come from about 90 species, and there are 250,000 at least species in the world, then common sense tells you there have to be more drugs in plants if we rely on only 90 of them to provide 25% of the medication in the United States.
Dr. Richard Schultes of Harvard University has spent more than 40 years searching for plants that might help realize this potential.
There are 500,000 species of plants in the world. My interest is purely medicinal, but I hope one day at least one of the plants that I have brought to the attention of science may give us a new medicine.
An ethnobotanist, Schultes specializes in the medical uses of plants in traditional cultures. His work has focused on the rainforests of the Amazon and in teaching others the ways of the jungle.
The blowgun among the peoples who use it is extremely accurate. They will put
Blowguns and poison arrows have played a significant role in Schultes' research, and he enjoys showing off his prowess with them.
Then, of course, I'm in a building here, and I can't shoot very high, but we're going to try to see if we can hit that target up there. When I got my PhD at Harvard in 1941, I had a grant to go down to work on arrow poisons in the headwaters of the Amazon. Arrow poisons, which the natives used to kill animals, were becoming very important in medicine.
The best known of those arrow poisons, curare, has become indispensable as a muscle relaxant during surgery. To Schultes, the Amazon was an obvious starting place for botanical research.
when you look at any flora, even our own depaupered flora here in New England and Canada, it's a veritable chemical factory. Multiply our 1,800, 1,900 species, multiply that to 80,000, and you can see what an emporium of chemical wealth the Amazon is.
Schultes himself has collected over 24,000 plant specimens, many of them previously unknown. With such plant riches available for study, botanists must find a way to focus their research on the plants most likely to have medicinal value.
If chemists are going to have to get material of and analyze 80,000 species, they'll never finish the job. One shortcut is to concentrate on those plants that the natives have found to have some activity on the human body.
Schultes found the local medicine men to be his most valuable tutors in plant medicines. He even participated in their rituals. He's the tall one on the right. Mark Plotkin, an ethnobotanist who was trained by Schultes, also works in the Amazon, using the same methods.
Well, according to Professor Schultes, the oldest profession in the world is actually that of the shaman or witch doctor. But through the course of human history, most of the world's medicines have come from plants. It's only relatively recently, with the advent of modern technology, that we've been able to reduce our dependence on medicinal plants as a source of medicines. The medicine man or shaman or witch doctor or brujo or curandero is the person who knows the most about these plants. Now, if you look in Western literature and movies and films and books, he's often been derided as somebody who runs around with rubber snakes, screaming gibberish and mumbo-jumbo. But the people who've had the real pleasure and the opportunity to work with these people has found out quite the opposite, that this is a very serious profession. This is the predecessor of modern doctors, and we have a lot to learn from these people.
This is Dionisio Aguinda. He is a brujo, or medicine man of the Ecuadorian Amazon. He is searching for plant remedies he can use in treating his patients. For Aguinda, a Quichua Indian, the rainforest is a living pharmacy, a potent, varied, and infinitely renewable source of plant medicines. Aguinda uses this bark as a treatment for pain. He draws on an extensive but unwritten body of knowledge about plant medicines and their preparation. For today's treatment, the bark is prepared according to a formula that calls for it to be boiled, along with several other ingredients. The formula, along with countless others, was passed down to him from his father and grandfather, who were also medicine men. His patients often journey for days through the jungle to seek his help. This woman has a persistent stomach pain. Because of the bark's unpleasant taste, the medication was prepared in a weak broth. Like many plant medicines, it must be taken several times to reach its full effect. The Western reaction to some of these medical practices is often a skeptical one.
One of the things you learn in working with these different cultures is these people have a different approach in many cases to curing and healing. And in the case of some Indian societies in South America, the medicine man or curandero actually takes the medicine, which in this case is a hallucinogen, and defines the cause of the illness and attempts to cure it through contact with the spirit world. There's a wonderful quote where it said the white man goes into his church and talks about Jesus, and the Indian goes into his teepee and talks to Jesus.
In ceremonies like this one, the patients entrust their health and their lives to an ancient alliance between the natural and the supernatural. There will often be further treatment with herbal medicines, and some of the hallucinogens themselves may prove to have important medical applications.
In the 1940s, Professor Schultes was working with Indians in the northwest Amazon, and he found them using the sap of a tree of the nutmeg family for two purposes. One is as a hallucinogenic snuff, and the second to paint on fungal infections of the skin. 40 years later, I was working on the Suriname-Brazil border, and I found the Indians using this for the same purpose, that is, to paint on fungal infections of the skin. Now, modern medicine cannot cure serious fungal infections of the skin. If you get a bad case of athlete's feet, for example, you have to suffer from it. We can only suppress it, we can't cure it. A real problem with cancer patients is because of the problems with their immune systems resulting from the disease or the medication, is they have problems with fungal infections of the skin. Now, a Brazilian plant chemist looked at fresh material of this family and found 3 new compounds previously unknown to science, 2 of which have antifungal properties. It's my belief that focusing on plants which are used for the same purpose by different peoples are where we're going to find new and important medicines for the future.
This remote part of Ecuador has recently become the focus of intensive scientific study, part of a systematic effort to document the elaborate medical tradition of the Quichua Indians. In a collaborative venture, ethnobotanists Robin Marles of the University of Illinois and David Neal of the Missouri Botanical Gardens head off on a collecting trip.
Our project is involved in basic biological inventory of the Amazon forest to find out just what's here, what plants are here. Because although this region, the Upper Napo, is one of the richest areas biologically in the world, it's very poorly known.
Today, Dionisio Aguinda allows the scientist to accompany him as he sets out on one of his routine trips to collect medicinal plants. This fieldwork is part of a massive effort to identify and investigate nearly 2,000 therapeutic plants used in the northwest Amazon. Even to experienced naturalists, the sheer diversity of the Amazon, with its more than 80,000 plant species, is overwhelming. Without Aguinda's guidance, their chances of discovering useful medicinal plants are remote. Aguinda's intimate knowledge of the jungle and its healing plants has been a family legacy for generations. But around the world, the knowledge of such natural healers is on the verge of disappearing. Aguinda's son, Enrico, represents the end of the family tradition. Instead of training as a medicine man like his father, Enrico has become a guide and translator for the growing traffic of prospectors, oilmen, and even tourists who now filter into the heart of the forest. When Aguinda dies, his family legacy will pass from the human record, and science will lose a vital link to the mysterious plant kingdom.
What we're looking at is a culture that's standing on the edge of a precipice, and this culture and this knowledge is going to disappear over the edge of that precipice unless we work to document that knowledge in the next generation. Every time one of these old medicine men dies, it's as if a library has burned down. In fact, it's worse than that. If a library in this country burns down, most of that knowledge is found in other libraries as well. When these old medicine men die, that knowledge is lost forever.
With an increasing sense of urgency, the research team will attempt to make a permanent record of Aguinda's medical knowledge. Following his leads, they continue their search for samples of the plants Aginda uses, which might have further medical potential.
This is a species of Sloania.
This fieldwork in Ecuador resulted in the collection of over 200 species. Before the scientists leave the jungle, they must prepare the plant specimens to be shipped back to the U.S. Through the ages, botanists have used similar methods to organize and preserve their findings. Samples of each plant, called voucher specimens, are first sorted according to common characteristics, such as leaf shape and structure or presence of flowers. The voucher specimens are then pressed between layers of newspaper to dry. Known species are identified on site. For other species, any special characteristics that might aid in identification are noted. Unfamiliar specimens may also be recorded by a botanical illustrator. In some cases, a plant will warrant more than a single specimen.
If it seems particularly interesting, for example, something that's used to treat intestinal parasites, or something that's used to treat sores which don't heal for a long time, then what I would like to do is to collect about 5 kilos dry weight of the plant to bring back to Chicago for chemical and biological analysis.
Plant chemist Dr. Bryce Douglas is familiar with the obstacles faced by collectors when large amounts of plant materials are required.
That is when it is extremely difficult, keeping in mind that very often the tropical conditions are 90 degrees Fahrenheit and 100% humidity. Try to dry natural materials, try to get the water out of them under those conditions. It's very difficult, and if you don't, they simply rot. So there are a lot of logistics of that sort that have made the chances of success just a little bit tougher.
Once the samples are back in the lab in Chicago, Robin Marles can begin the detailed chemical analysis of the team's collected specimens. It may be months or even years before they know if they've discovered anything.
You're only going to find activity in a few percent of all of the plant extracts that you test. Then from there, you've only got the initial stage, which is some sort of a crude extract or gum or goo that has the biological activity you're after. You've then got to go into the chemical side and start purifying that until you can get it down to a pure compound. That takes an awful lot of time and work, and at each stage, you've got to follow it by biological assays looking for the same activity that's desired in the end, so as to make sure that you're on the track of the active substance.
One active substance of potential pharmacological value has recently been detected at the University of British Columbia. Here, scientists have found unusual and highly toxic chemicals in a variety of garden marigolds. The marigolds contain compounds called polyacetylenes, which seem to be showing some promise in lab experiments as anti-tumor agents. In this demonstration, an extract from the marigold is added in equal parts to 2 dishes containing mosquito larvae. The larvae absorb the polyacetylene molecules into their bodies. These molecules are unusual because they are photosensitive, becoming active only in the presence of sunlight. The specimens are placed in an ultraviolet light chamber. One dish is covered so that its contents are shielded. The combination of polyacetylenes and ultraviolet light causes a violent reaction that destroys the cells of larvae in the unshielded dish. Those in the covered dish remain unharmed. Researchers are hopeful that such light-activated toxins could be modified for use in the human body, where they would be targeted to destroy particular cancer cells while leaving the body's normal cells intact. Extensive research into anti-cancer drugs that could be derived from plants is now being sponsored by the National Cancer Institute. It's the largest program of its kind in the U.S.
We've screened something over 35,000 plants, different species, over 120,000 extracts, and when you're looking at a large variety of substances with biological activities, you're going to find some that have good medicinal activity.
Thanks to the development of new technologies, the NCI scientists have become increasingly sophisticated in their approach to screening for anti-cancer agents in plants.
After perhaps 25 to 30 years of testing, we've now realized that we're not apt to find a kind of magic bullet, as it were, for cancer, because cancer is so many different diseases, and each cancer in each different organ is different to the others and doesn't respond to the same sorts of drugs. We're going to look for agents which are highly selective for specific types of cancer. So we'll be screening for agents against lung cancer, colon cancer, breast cancer, and looking for agents which affect those specific types of cells and not other cells in the body.
The Natural Products Division of the NCI is extending its search for new anti-cancer agents beyond the plant kingdom. Recently, they've carried their exploration for natural medicines to the sea. One promising discovery comes not from a plant, but from a marine animal called a tunicate, or sea squirt. Dr. Kenneth Reinhardt.
The sea is really a rather harsh environment in the sense that everything grows on everything else and everything eats everything else. So that something that's just sitting there, like sponges or tunicates, really have to have some way of defending themselves. The tunicates are especially interesting creatures. They are just sitting there, and they are invertebrates, like sponges. They don't have the shells that mollusks have, and they don't have the spines that sea urchins have, so they have to defend themselves chemically.
This chemical defense system is proving to be a rich source of potential new medicines. Among the several hundred species so far discovered, nearly 30% have been found to contain substances that destroy viruses or tumors, and the research is only just beginning.
Marine animals have really been very, very little investigated as potential sources of pharmaceuticals. This area has really expanded tremendously in the past 5 to 10 years, and there are many chemists and biologists around the world looking at new marine organisms as sources of pharmaceutical substances.
We're really very excited about the prospects for marine natural products. We've only been looking for new drugs from the marine area in the last 10 years. So in that time, we're really very pleased that out of our efforts, that one drug is actually in clinical trials now, Didemnin B, as an anti-cancer agent.
Didemnin B was the first cancer drug in the NCI program obtained from a marine organism. Didemnin destroys tumor cells by inhibiting protein synthesis. In laboratory tests, mice with leukemia that were given Didemnin B lived twice as long as those that were not treated. The drug is now in human toxicity trials. Didemnin B was first discovered in 1978. A complex series of steps, taking nearly a decade, were required to transform the natural chemical found in a marine animal to a refined substance that may someday save lives.
The substances that we get from nature are very often not the optimal substance which is going to be the final drug. But what we are looking for is all these wild leads, these unusual, wonderful compounds that we would never dream up ourselves. Sometimes they can go through to be the drug themselves, but most of the time you've got to take that compound and then either modify it synthetically or make related sorts of compounds by synthesis.
Many promising drugs must first be chemically altered to reduce their toxic side effects. This long, expensive, and painstaking process has been considered routine when working with substances from nature. But recent breakthroughs in computer technology, developed at the National Institutes of Health, may dramatically speed up this work. The process is called molecular modeling. Using this powerful computer system, scientists can simulate the interaction of a natural drug with human biology. By studying how molecules fit together, the scientists have a front-row view of organic chemistry that can never otherwise be seen, even with the most powerful microscope. The researchers use electronic puppet strings to simulate chemical experiments and can quickly determine which parts of a natural substance might cause toxic reactions. Their ability to visualize these experiments represents a dramatic advance. The process was pioneered by Richard Feldman.
On the one hand, it provides scientists with knowledge about the structure of proteins and DNA, and from this they can understand how the drugs relate to those things. But on the other hand, we can also do modeling by ourselves and then go to the scientists and say, look, you know, this is interesting, this is important. Why don't you start thinking about these sorts of problems?
Using this technique, the molecules can be manipulated and tailored so that side effects are minimized or eliminated. The resulting molecule becomes a template that the scientists can use to design a safe and effective drug. The combination of molecular modeling and new bioengineering techniques suggests a promising future for drugs from natural sources. But today, the major research thrust into natural medicines comes from academia and government, not from the pharmaceutical companies.
There are no more than 2 pharmaceutical companies in the United States that have research programs designed to look for new drugs and plants. It's a mystery as to why this is true, because in 1981, the American public paid $8 billion for prescriptions dispensed from community drugstores that have as their active ingredients drugs that are still extracted from plants. So you have an $8 billion plus market with less than $200,000 of research and development. So how can anybody expect a lot of new drugs to come from plants with this small emphasis on the discovery aspect?
One explanation for this might be found in the experiences of the pharmaceutical company SmithKline and French. During the mid-1950s, the company began an extensive plant screening program. Its director was Dr. Bryce Douglas.
We had felt for many years that there was a great potential in natural products which had not yet been realized, because we were aware of history, that so many of the then used drugs, this is some 20 years ago, the drugs then used had come from natural products, and particularly from a class of substances called alkaloids. So we systematically started looking at plants from their possible alkaloid content. Alkaloids are nice, identifiable chemicals. They have a little handle on them that you can grab hold of chemically, and so you know when one is present or not in a plant.
Using the techniques available at the time, the SKF team mounted the most extensive plant medicine research program ever undertaken, collecting and screening over 30,000 plants from all over the world.
The SmithKline and French program was a very systematic one, in which as much science as was available on the alkaloid side was utilized, both in finding the alkaloid, identifying its structure, and determining its pharmacology. What we found after looking at so many plants was that there was a uniformity of action in many of these alkaloids, unfortunately.
In other words, there was not enough new activity in these results to justify continuing a large-scale survey of plant materials. The logistics of collecting, ensuring a continuous supply of raw materials from the jungle, had become overwhelming. And there was yet another consideration.
There was something else happening at this time. The science of chemistry, the science of chromatography, the science of separation of natural materials, all was coming along. Advances in science always seem to happen in spurts. These things happened, and so this helped indeed with the logistics. If you once isolated the alkaloid material in its pure form, it now became possible through X-ray crystallographic analysis to obtain the chemical structure of a complex substance in a reasonably short time. That helped a great deal, in fact, to move natural products along, but it also helped to move us away from the natural products method of screening and collection, because now you had a chemical structure that you could manipulate by the very powerful organic chemistry of the day. And indeed, that's usually what happened. A natural product was the focal point for the start of a very intensive effort to modify a structure to produce the medicine. So natural products are really always with us, but in the formal sense of collecting, the logistics became burdensome for SmithKline by about 1966.
The program was formally ended, and SmithKline, like other pharmaceutical companies, began to direct its energies toward the chemical synthesis of new drugs. But now, 20 years later, the research emphasis may be shifting back again.
I think we're seeing a change in attitudes, and we're going to see much more of this type of sponsorship in the future with modern technology and the ability to analyze plants in a much more rapid manner than we were in the past. I think this is the wave of the future. And pharmaceutical companies in Western Europe and in the third world in particular are very interested in plant medicines.
It has been said that there is a preference by pharmaceutical companies for a purely synthetic approach to discovery of new drugs and over the natural products. In fact, these two worlds are not separate and they link very much together. I think no matter how brilliant the chemist or synthetic chemist, we're all dependent on the natural ideas. So I don't think it's one or the other. Each area takes its predominance based on the point of evolution of the sciences. Today, in fact, we're much more deeply back in natural products than we ever were with genetic engineering. Because now you can, in fact, obtain a useful result with a small amount of a natural material, and instead of heading back to the jungle to collect it in large amounts, you now head to the molecular biologist's laboratory, and he will probably produce it in a yeast for you, an uncommon host for that kind of material. So in fact, we're back more deeply in natural products than ever, and all the pharmaceutical companies obviously are, many institutions are.
The medicines of the future are likely to come from the chemical modifications to nature made possible by new bioengineering techniques. But the most important idea from nature, the plant gene itself, can't be synthesized.
I have a real problem with the fact that most of the general public thinks that genetic engineering is going to save the world. They think that with genetic engineering, we can invent everything in the laboratory. Nothing could be further from the truth. What genetic engineering allows us to do is combine different genes in the laboratory. This will have great use in the future if we preserve many of these genes, which are in great danger in the tropical countries.
The tropical rainforest, nature's great storehouse of plant material, is under threat. Each year, over 28 million acres of rainforest, an area about the size of West Virginia, are destroyed in lumber and land development projects. In the Amazon basin alone, over 40 million acres of forest have been cleared. But the thin soils that underlie these Amazon forests can sustain crops or cattle for no more than 8 years. Roughly half of this area is now abandoned, and the soil and plant life it once supported cannot recover.
It's been estimated that about 10% of all plant species on the face of the earth will become extinct per year. For every plant, therefore, that goes extinct, you have to consider it also as a potential source of a new cure for something. And so it has terrible implications.
If we don't increase our commitment to conservation, we're going to see the rate of species extinction increase rapidly. Species are going extinct. Each time one of these species goes extinct, we're losing something. In most cases, we know not what. Who knows if it's a cure for AIDS? Who knows if it's a cure for cancer? Who knows if it's a cure for other afflictions that will develop in the future? When we lose these things, they're lost forever.
Awareness of what depletion of the world's biological diversity would mean has inspired some strategies for preservation. These exotic plants have survived from prehistoric origins in southern Africa. Today, like many plants in the Amazon, they are on the brink of extinction. But these species may still be saved. At the University of California's Arboretum, these plants and other rare and endangered species are protected. Under the direction of Dr. Harold Kupowitz, the Arboretum is a botanical ark and may represent the last chance at survival for many plants. An authority on plant extinction, Dr. Kupowitz is committed to preserving endangered species, their seeds, and their pollen. According to Kupowitz, the loss of 10% of the world's plants, as many as 70,000 species, would threaten a whole range of wildlife, from insects to songbirds, and be an unimaginable loss to modern medicine.
We don't know what good genes there are in particular plants, and we can't foretell when we're going to need them. But if we don't do something to save as many as possible, we're potentially impoverishing ourselves and the future society as well.
At the Arboretum, each species is viewed as a precious record of genetic information passed along in the seeds of the plant. For each plant here, a makeshift gene bank contains their genetic records.
What I've done here is taken out 2 samples of seeds. Each tube has about 50 seeds in it, and they're sealed in a glass test tube.
If a plant becomes extinct, as some here already have, these fragile records may well be all that remain.
Once it's frozen, the seed can stay in a state of suspended animation literally for centuries. It can be retrieved in the future by anybody else who wants to see what that plant was like or to investigate any of its properties.
The gene bank, although an important effort, cannot substitute for genetic diversity in the wild.
These technological solutions, like cryogenic gene banks, should not be thought of as alternatives to conservation. We really do need to conserve the forests and the environment, but we should think of the gene banks as fail-safe devices so that we can retrieve plants from them if the plants we need are lost and do go extinct in the wild.
Because of the realization that conservation is about protecting life on Earth and protecting many of the plants and animals that we depend on, people are seeking new and innovative means of protecting biological diversity. For example, in a type of approach being pioneered by organizations like the World Wildlife Fund or Conservation International is the so-called debt trading approach, buying up third world debt and allowing them to use the money to spend on conservation approaches in their own country.
In just such a Debt for Nature program, Conservation International has recently bought up $650,000 of Bolivia's massive foreign debt in exchange for setting aside 3.7 million acres of protected rainforest. Other economic incentives for conservation have also had some success. Carefully managed forest areas in Thailand and Nepal currently account for over $1 million a year in medicinal plants and related product exports. But given current projections, it is unrealistic to expect that rainforest destruction will cease, or that the plant kingdom can be entirely preserved. In a race to discover and understand what still exists, scientists in the field continue their search for the hidden power of plants. In his office at Harvard, Dr. Richard Schultes pursues his lifelong quest for new medicines from among the thousands of specimens in his collection. And if there is a wonder drug in these dried leaves, we can only hope that the plant it came from can still be found.
I think we have to keep in mind how little we really know about creating certain chemicals. I think it would be arrogant of us to feel that we can do and invent and synthesize everything in the laboratory. I mean, we have to remember that not only has Mother Nature synthesized many more chemicals than we have, Mother Nature synthesized us as well.
For a transcript of this program, send $4 to Nova, Box 322, Boston, Massachusetts, 02134. Please be sure to include the show title. Major funding for Nova is provided by this station and other public television stations nationwide. Additional funding was provided by Allied Signal, a technology leader in aerospace, electronics, automotive products, and engineered materials. And by the Johnson & Johnson family of companies, supplying healthcare products worldwide. A companion book, Nova Adventures in Science, published by WGBH and the Addison Wesley Publishing Company, is available in libraries and bookstores nationwide.